rain shadow or the shadow effect off the Olympics is so so specific and so variable, right? Where we both live, that like 5-degree change in wind can totally change the weather that you're having versus what I'm having.
Absolutely. Even around the South Island is still,
Mm-hmm.
it changes drastically within like a 20-minute drive.
Mm-hmm.
20-minute drive, you're like, this place regularly gets snow every single year.
Yeah.
Yeah, the place, like, we'll go years without it.
And the ferry, so Sydney is where you get off the ferry if you're going to Victoria, BC, Canada, which is a beautiful place to visit. Highly recommend it to everybody watching on Nautilus Live. Someone described, the last time I was in Sydney meeting Nautilus there, someone was like, this is the Florida of Canada. And because Sydney is even warmer than Victoria is, because it's kind of tucked around on the inside of the Salish Sea. There's lots of folks who retire to that area because they don't want to shovel snow anymore, and they want a more moderate climate, which makes me laugh.
I used to call it the Florida of Canada, but I've changed recently to the Hawaii of Canada because it's an island.
It's true.
And it's very Hawaiian.
It's okay. Okay.
There's some other comparisons that made it seem pretty good. Anyway.
Is it volcanic?
No.
I think it's an exotic, an exotic terrain. It's just a piece of continent slapped onto it from a subduction process.
It's good surfing.
Oh, that's cool.
So exotic and Hawaiian kind of sounds consistent. I love the gardens there, though. They're just gorgeous.
Butchart?
Yeah.
I go there most years at Christmas time. They decorate the whole thing with lights and stuff. It's lovely.
I'd love to see it at Christmas. I
The
haven't
outdoor
seen it
skating
then.
rink and
So
serve
cute.
hot chocolate.
That's awesome. Here you go. Here's a fun fact about plow steel.
Yes.
Plow steel is a high-strength steel which contains between 0.5 and 0.95% carbon. Improved plow steel.
Oh, this is. Oh, I know that. Okay, go on.
No,
sorry.
go for it.
The 6.8 is the double improved plow
Double
steel.
extra improved.
Extra improved.
Extra
That's
improved
extra
plow
improved
steel.
Plow steel. Yeah.
Extra improved plow steel has a minimum braking strength 15% higher than improved plow steel, which is
I heard that now.
stronger, more durable, and more wear resistant than normal plow steel. So we're talking like Olympic podium here, where extra improved plow steel is right on top.
So is it all in one brand? It's just their different, strengths. Like premium plastic wrap is better than,
I think so. Yeah.
store brand.
Yeah, I think it's. I don't know if it's branded though. Like I'm not on a branded site here.
No, I
as
think
I'm
it's a
reading
metallurgical
this.
term.
Yeah.
Oh, okay.
Extra improved plow steel has a tensile strength of 1,960 Newton per square millimeter, or tensile strength of about 285,000 pounds per square inch. That's a lot of pounds, and we have a lot of square inches.
Well, a lot of square inches. I don't know the cross-sectional area of the plow steel in the 6.8.
Yeah.
You can do the math of the cross-section of the entire cable, but I don't know how that relates to the specifically the
We
armor.
have. Yeah, I was thinking we had a section of the 6.8, but we don't. We have a section of the ONC
The
cable and the tether down below.
Where are the corals, Steve? Where'd they go?
Excuse me, corals. Where are you?
Do you have a
Your
coral
sound.
call you can do?
What would the call? I feel like it would be some kind of like sifting noise just from all the polyps like sneaking things off.
I actually make bird calls.
Do you?
Yeah.
What?
Yeah. What I do is probably the best one I do is the something called the mute swan. Want to hear it?
Yeah.
Oh, no.
You want to hear it again?
No.
Yeah, that's good.
Let's do this the whole way up to the surface. This is the best.
Blue water is going to be great.
It is. That'd be really great.
I wish our listeners could hear me looking at you right now.
Yeah, yeah. What would the sound
totally
be
can.
for the like type of glare I'm getting at this moment?
Hey, you see that? You see that sponge in the upper right?
Upper right sponge, the dark one?
Yeah.
I do see it. Let's check it out. Let's check it out more in depth. And
So
yeah.
Do we see that urchin?
Wait, the dark one or the white one?
The white one.
White
yeah.
one's a sponge. Dark one's an urchin.
12 o'clock.
Yeah. Go ahead and zoom.
Zooming.
Looks like a kerosene lantern.
Mm-hmm.
Or an oil lamp or something.
Tell me about it, Steve.
Yeah. So we tried to sample one of these on our last dive and didn't quite succeed.
Okay.
It's in the genus Poliopogon, I think. And I've been looking for a good opportunity to sample one, and
This is that.
this is that.
Do you want to break a piece off?
Yeah,
maybe?
like
and
a-
slurp.
A chunk and slurp. Yeah.
Let's try it.
It
Come wide, please.
floated out twice.
Coming wide.
I will have to do it.
When
not
we sampled it
total
yesterday,
casual,
so
We
full
sample it?
wide.
Mm-hmm.
so I'd like a chunk and slurp,
Full wide.
and it's gonna be the consistency of cotton candy, not
Cotton candy consistency.
crunchy.
Crunchy cotton candy consistency.
No, yeah, like soft cotton candy.
Not,
no
not
crunch.
crunch.
Yeah.
Squish,
crunch.
squish, not crunch.
Yes.
Squish, not crunch.
Yeah.
In the
Super
Dr.
light. Yeah.
Gabazoy English system of phylogeny.
Could you get my camera set up for the slurp, please?
Oh.
So switch that one over to the bucket cam.
Yeah.
Push in halfway, please, Wayne.
Okay.
Doesn't have to be huge, but something that'll fit in the slurp.
Okay, good there, Wayne.
Okay.
Oh, wow.
That is not
Wait,
crunch.
he's squishy.
Yeah.
Why is he squishy?
I don't think Miles is listening to SBO. We just had a whole conversation about him being
Okay, come
squishy,
wide, please.
not crunchy.
Coming wide. Oh, do you want a close up or?
It's up to science. You want a close up?
Yeah, if possible, please.
Okay, and can you set me up with a bucket? What was the bucket you want, Suna?
2, 3, or 4.
Okay. Go ahead, zoom, Wayne.
Zooming in. Getting it in focus.
Is that on 4 or is that?
That's almost on 4. That's all right. Okay. You good, Suna?
Yep, thanks.
Okay, come wide, Wayne.
Coming wide
That's focus.
with the other one.
All right, and you can turn the suction on to 50%, I think is what we'll need here.
All right, and on set 3. Uh-oh.
Did it get stuck?
Need a plunger.
Yeah.
There we
There he
go.
goes.
And as soon as that
And
comes in the jar, you
Yep.
can turn off the suction.
it's don't.
There we go.
There he is.
Got it.
Which
Thank
jar
you.
was it?
4?
4?
Yeah.
Thanks.
All right. Loving the efficiency in this last hour.
And flush at your leisure.
Okay.
Oh, that's really cool.
Okay.
You got one of those
And
swimming
come up and
sea
you don't-
cucumbers.
Yeah, we
Yep.
actually sampled this same type of bamboo. This is the type of bamboo that was covering that other peak.
Huh.
And yeah, it's crazy dense bamboo coral colonies by this standard.
Mm-hmm.
Correct.
And we even found one that was covered with zoanthids that we sampled.
Mm-hmm.
It may be a gold coral,
Yeah.
but we got a branch to identify it, both the bamboo and the zoanthid.
Very cool.
Mm-hmm.
Yep. Yeah.
Yeah, the spatial variability of the coral are really amazing.
Yeah, you could say you could almost spend a whole dissertation understanding why the corals occur where they do.
Almost say.
Almost, yeah.
Or several.
Probably several, yeah.
Yeah.
Because you have a couple scales of scales of things going on here. You have landscape scale, where over one feature you have different zonation patterns based on what species are there, and the micro structure in the rock, what they can attach onto, slope, habitat, micro habitats like cobble-sized rock or boulder or pebble, pavement, and then you have the biogeographic, scale that deals in kilometers or tens of kilometers, hundreds of kilometers.
It's so interesting. I mean, I admire the process where you just kind of throw ideas on paper. The idea that there are these distinct biogeographic regions across the Pacific, but then when you start looking, that isn't always a perfect explanation of what's happening. There's mixing and one big ocean in a lot of ways, but then also incredibly distinct just on the layers patterns you want to look for.
And I'm just trying to draw parallels to like a floral progression on land. And it seems like so different. You don't have that natural going from, pines to hardwoods or anything like that. Or softwoods to hardwoods here. Or do you, Steve?
I don't think it's the same way. So I'm reading a book, or have been reading a book, or trying to been reading a book for.
There's a book in my room.
Yeah, there's a book in my room. Occasionally it's on my desk. Occasionally it's on my bedstand.
Love it.
It seems to walk around.
Yeah. About Humboldt. Does anyone know about Humboldt?
Tell us.
Alexander Humboldt?
Yeah. He in the early 1800s, late 1700s, he did some exploration around Central America and South America, and he basically mapped the distribution patterns of plants on gradients up mountains, and he was famous for doing these maps of like Gran Canary Island, the volcano, and the vegetation on Gran Canary, and a few of the mountains in the Andes, northern Andes, and he made these great maps later in his life about where these break patterns occur, and I think we're kind of doing something similar in the deep sea, but we're still trying to understand what those gradients are. It's not necessarily altitude, or temperature may be similar, although temperature is going to vary probably much greater in air than in seawater.
Not a precipitation issue.
Yeah.
Yeah.
You know what the book's called?
Yeah, I can show it to you. I have it. It's called
like the measurement of the
Invention of Nature, I think it's called. Yeah. It came out a couple years ago. I bought it when it came out, and
That one's been on my list.
I read it. I've been reading it on long flights. I should have probably gotten the audiobook. I would have been done by now, but yeah.
Let's look at this purpley thing. Okay, Wayne, go ahead and zoom.
Zooming in.
So we have
Cool.
a
Yeah, you can see his
sea
mouth.
cucumber.
Count Chocula sea cucumber.
Well, that's one of the first ones we've seen.
I'm not sure that's.
No, I mean
yeah,
on in this area.
Yeah.
Yeah.
Hello. It's smiling.
Oh,
Is that his mouth?
I think those are the mouth parts. but wait and see.
We might
I
find
think
out.
that's called mooning.
Yeah, those are the mouth parts.
Yeah, you can see the two-peter like tucked right
Mhm.
inside.
Oh,
That's
yeah,
impressive.
Oh, yeah,
There.
he's
he's
doing some ballast management there.
yep.
Yeah.
Ballast management.
Yeah.
Yeah. And now he's
Right
like,
there.
Look at me go.
and I'm out.
Oh, that's great.
So flexible.
Yoga.
Yeah, I mean that's yoga hashtag
Big
yoga goals
stretch.
right there.
Yeah.
Yoga every day.
Oh, we're really close. Yoga goals.
It makes
How
me,
is that a method of propulsion? That is
isn't it
wild, isn't
cool?
It's so cool.
Yeah.
It's like touch your toes with a back bend.
Touch your toes over your back. Yeah.
Okay, coming wide.
Do we know the species of this cucumber?
We probably have a genus.
Oh, it's
I
so
think it was
cool.
Benthothuria. I'll write it in the chat, but it's tentative ID. Very nice.
Okay.
That's
So
a
I-
great shot.
Yeah, I
Even
remember
though we
how
get to see these
fascinating
often,
the.
these are so cool.
Turn on the porch light,
I
please.
remember how fascinated everyone was with the
Is he in bubble?
Inypniastes in the Gulf and the Caribbean, the headless chicken fish.
That's full wide.
This is a pretty close relative.
Wow.
I don't
hello.
necessarily not fond of the headless chicken fish analogy, but I can see why people call it that.
Does it just make you uncomfortable that it looks like a headless chicken fish?
Yeah, I never got it. You know.
I mean, I really get it
All right.
in like a couple
It's
still
showing
shots. It's
off
great in
now.
bubble, yeah. Which is on the quad channel if you're watching home.
So the mouth parts that were on the top end, those are kind of like suction discs it uses to, yeah, like you were saying the other night.
Mm-hmm.
Yeah, I should move along.
What was that analogy? Using your fist to
Okay.
smash
Yeah,
your
if-
food into your mouth?
Yeah, if you just like smashed your palm down onto your plate
Yeah.
and the palm
Thanks,
of your hand was sticky.
Yeah, Velcro.
Yeah, and then you just put it up into your mouth and
Yep.
I don't
What
know.
was that? Putting your fist in your mouth? Sorry, I missed that. What are you talking about?
No, it's how sea cucumbers eat. They have sticky pads, mouth parts. So like if you took your breakfast and you just smashed the palm of your hand down and your palm was sticky and then you put it up to your mouth and ate it off while you did that over and over again.
Huh.
Really great way to get uninvited to family holidays.
Right.
Impress your cousins or your kids. Any young people you know.
It's family
think,
holiday. You can get uninvited.
Absolutely.
If you work hard enough, yeah.
Maybe teach your nieces and nephews bad tricks.
I was explaining how not sea cucumbers, but sea stars eat at Thanksgiving is why I bring it up. And my cousins were not that impressed that I take your whole stomach out from your mouth and then wrap it around your plate, digest it all outside your body, and pull the goo and your stomach back inside of your body.
Are you asking if I'm on SPL? Yes. Is that okay?
That's great. Don't leave.
So
No, I'm unquiet.
yeah, the sea cucumber, nice.
Yeah, this looks like a sheet flow here. This is where we're getting a lot of our coral last night on top of the sheet flow like this.
Yeah, I'm seeing a lot of unbranched colonies. We've sampled at least two of them. The two different types that seem to dominate these slopes are a thin, unbranched primnoid octocoral, which you guys slurped at the beginning of this watch change, and then a bamboo coral, sometimes has curlicues in it.
Did you see the many black corals at one mound? I don't know. I think it was the previous or two.
It was the second seamount we went up.
We sampled,
Second, yeah.
we sampled one
they
of it.
Yeah.
never
Yeah.
left it.
I did see, and I can't identify it yet.
Yeah.
I didn't get a good chance to look closely, but that is a good collection. Kudos.
We thought it might be representative because
Yeah.
it was everywhere.
Yeah, that's great.
Oh, okay, you mean the lighter colored one.
Yeah.
Yeah, that was the first 2 peaks we went up. There was everywhere. Yeah.
It was the second peak, I think, right? Not the first.
Well, we actually saw it on the first peak quite a bit too, and that's when you said maybe we should get some of these.
It's about time.
Yeah, then we saw a bunch of them on the second, the same.
Yeah, that's a great move, is to get things that are representative, because there's nothing more frustrating than doing video analyses and having your top species contributor being unknown bamboo, unknown bamboo coral or unknown black coral species A.
You hear that?
Traditions.
I mean, people are used to it when they study deep sea systems, that you might not have all the identifications you need, but at least it's distinct, and even better if it's a sample. There's something to the right.
To the right.
Anemone.
We can have a look at that one.
Yep. Snap in. I think it's something we've seen before. But
I think it's the Syrian.
Okay, Wayne, go ahead.
Zooming in.
the name of the anemone?
Do you think it's a Serianthid?
It's not a Serianthid. So Serianthids, the way you can tell Serianthids from other anemones, this actually might be a Corallimorpharian, based on the broadness of the oral disc and the knobby tentacles. But the Serianthids are really distinct because, so they have an outer row of tentacles, but they also, right around the mouth, have a second row of tentacles.
And much thinner tentacles too, right?
Yeah, the second row is much finer, yeah.
Okay, come wide,
Very
please.
nice, thank you.
Wide.
I got derhubarbed.
I used to have a pet anemone.
A pet anemone?
Yeah.
Wow.
It was one that was collected during a diving trip, and I had it in a flow-through tank in the previous lab I was at, and feed it bits of mussel. I would grab onto it, ingest it, come back a few days later, feed it some more.
Wasn't a deep sea anemone?
No, it was a shallow. You know, I think it was maybe found at 30 meters.
What was its name?
I did have a name for it, but I forgot.
Did it wag its tentacles when you came home from work?
No, not so much. I think that was the only pet I've ever had.
That's a fun fact about you, Steve. Never have a pet? Except for that?
Yeah, it wasn't for a lack of trying. I just, never got around to it.
Fish. It wasn't Wolfgang and Enemy Mozart?
Step outside.
You're excused. Thank you.
Well, I had two birds growing up. One was Zygmunt Boyd, and the other was Ferdinand Magellan.
That's amazing.
Sigmund Boyd.
No, it's not.
Ferdinand Magellan.
Wayne does not
made my
think it.
absolute day. What kind of bird was Ferdinand Magellan?
It was a lovebird.
No.
Well, what do we have here?
Wayne's like, I would like everyone to look at the seafloor.
Hey, let's zoom in on
Back
this.
to the seafloor.
Sure, Rain, I'll throw you a bone. Spongeman Freud.
Also, a shout out to the Okeanos team who just had a googly eye stubby squid on their expedition.
Oh, right on.
Yeah, at Blakely Escarpment. So that's awesome.
Okay,
Thanks
coming
viewers
wide.
for letting us know. Tuned in, watching both feeds.
So fun fact, my colleague and lab mate, Alexis Weinig, is going to be out here for NA114 in the Howland and Baker and Johnston cruise. She's the co-lead scientist on Okeanos right now.
Oh, that's awesome.
Yeah.
Well, congratulations, Alexis.
Yeah. They're doing a great job. I'm getting dive plans, and I'm so bummed that I get all these reports about what they're seeing, and I can't watch them. Because usually in my office I have both screens or 2 or 3 screens going at the same time.
Yeah.
With all the different live feeds.
Yeah, this week was wild. There were four ships all broadcasting last week.
And what 4 ships were those?
Us, Okeanos, Falcor is out on the Cascadia margin right now with several scientists we worked with on the Cascadia margin. Was it Aleutian or is it? It was
Atlantis.
It was Atlantis doing OOI work.
Ocean X is out there looking at skill sharks in the Caribbean.
Slime Star, dead center.
Cool.
Yeah, let's zoom in. Zooming.
So is that 5 ships then?
Is it?
Maybe Ocean X wasn't coming.
Yes, it is.
Yeah, the Interspace Center doesn't take Ocean X's feeds, so 4 through there, which is where the tweets really took off.
Where it matters.
No. No.
All right, come wide.
Coming wide.
Very nice. So like hagfish, these are called slime stars because if you put them in a bucket, yeah, they'll slime it up.
Slime it up.
The Paragorgia too, we were sampling it, processing it, and they produce a lot of mucus too when they're stressed.
Oh yeah, I remember.
Bamboo corals can too sometimes.
Yeah, the filamentous stuff is a little more obvious now. The sedimentation goes down.
Mm-hmm. Oh, let's see a small branch on the rock that, but I think we can skip that one. I think it was a primnoid, one that we sampled. There might be a tube anemone in that sediment channel there, lower right.
Oh yeah, sorry, I gotta move a little bit.
That's
Let me
fine.
get some in the bank so I can stop better.
Yeah, we're definitely getting up to a more abundant biology umbellula sea pen in that sediment there. More umbellula.
Where do you see that?
Right
Okay,
there.
that little bit, okay.
Yeah, they can't quite see the stalk usually, but you can usually see that tuft of polyps.
Okay, I got some. I'm a little ahead of Argus now, so we can do more zooms.
That's when nothing shows up.
Yeah.
Can we pan right?
Yeah. Oh, look at this
What
thing.
is that?
Bamboo.
Bamboo coral, yeah.
Wow.
I saw
Dead?
the base of the corner of my eye.
Not dead.
Not
I can't
dead.
tell.
Not dead yet.
Oh no, there it is. All right, zoom in, please.
Oh, okay.
Yeah, these seem to be common in this area at this depth. I recall it was sampled in a few different capstone regions, as well as in the Phoenix Islands. We have a bunch of this. I'm still trying to identify this thing from collections from 2 years ago, and difficult.
All right, come wide.
Difficult morphology. But I haven't been paying as much attention to the bamboos as I have to other families that are a little bit easier to identify.
How are bamboos ultimately identified? What are some of their features?
So the morphological characters are: does it branch? Does it have branches? Where are the branches relative to the nodes? What is the shape of the polyp?
There's
when
a
it's
red
closed
fish.
or open?
Oh, shrimp, yeah.
Oh, it's a shrimp.
What is the sclerite structure in the body wall of the polyp? Does it have what are called intertentacular sclerites, which are spines that come out between the tentacles when it's closed up? Other things. Yeah, shape of the polyp. Sometimes thickness of the tissue. Is there any wash? Like, does it have a yellow wash? Sometimes can be used to identify certain clades. We saw a bunch of the yellow wash type bamboo corals earlier in the dive.
Sea pen over there.
Yep, Umbellula. Yeah, they're sediment dwellers. You won't find them on the rocks, so there's enough sediment there for them to dig in at least a few centimeters.
Peduncle in.
Yeah. I think it's one of my favorite corals to say.
Umbellula.
Although Kula Manamana is also pretty cool.
That's good. I like metallic gorgia.
Yeah. Yeah, so Metallogorgia is interesting. There's like one worldwide species, Melanotrichos, that is pretty common. It's in Atlantic and the Pacific. It might be elsewhere as well. But there are other species in the Pacific, especially the Western and North Pacific, that we know about, that are not as well documented because they've been rarely collected. And I think I have one of those in the lab right now. I'm trying to refine the identification for, because it's genetically distinct, and we have multiple specimens to confirm that from Melanotrichos, which is the...
Let's look at this thing.
Yeah.
You go ahead and zoom on this little purpley thing.
Zooming in.
The slimy.
Slime star.
Another slime star? Oh, yeah. Another chicken skin.
Look like a little dumpling.
Yeah, is there something on that? Like a... Or is that just a fold of skin?
Can't
Huh. I think it just looks like part of the animal. All right.
Bye-bye,
Oh, wow.
slime star.
You were flying in while
I
I was zooming.
I usually do that. Get you to zoom before I'm actually there.
Well, that's super exciting that you think you have the other Metallogorgia species like in hand in the lab.
I think it's been described, but there's only ever been 2 collections in the world of that type. Yeah, one is in Paris, and the other is in New Zealand.
And one is in Philadelphia in your lab.
Yeah, 2 in fact.
Philly.
I think you need to go there and see them.
Yeah.
Love to.
That seems like it should be a budget item. Coral tourism, getting to go see them. I guess that's what you're doing right now. You're getting to go see corals.
This is the most extreme form of coral tourism.
New industry, coral tourism.
Yeah. Some people pay to go to the International Space Station, and we paid it with our time to go to the bottom of the ocean.
None of me.
The fact that this is our job is pretty ridiculous. It's so cool.
Well, making good time. I didn't even think we'd make it up this far. But we've got, how much time left on bottom?
18 minutes.
15 minutes.
All right.
Nice work, Trevor. Making good time. Hey to everybody that's here joining us. Thanks for sticking with us through the last part of this dive. Turkey and Martinique and Bonaire and Barbados. Australia, Austria's here. Denmark, Finland. Hi in Japan and the Netherlands and Norway. Germany, Turkey, Canada, the UK, and the United States. Send in your questions. Let us know what you want to know about while we're here on the seafloor, or what you want to know about our team while we're wrapping up this dive.
While we're waiting, can we look at the yellow coral on the right?
Yeah, absolutely.
Always good to check. There's a lot of different yellow corals, and as far as I can tell, we're all dealing with the same species. I haven't seen too much morphological differences.
Plexaurids?
Yeah,
yes.
I would say so.
They're so hard when you get them on
Was
that
that
angle.
a stalk mushroom next to it? Did you see that?
Oh, it looks
to the left or right, or where are we?
To the left.
I think it's an umbellula sea pen.
There's the sediment.
All right.
Coming wide.
Sure. Is
how
it a
far?
is it a white one?
Oh, the little clear thing.
Oh, that thing.
Oh, that? I didn't see that. No, that might be a
little sponge?
Small sponge. Yeah.
Okay, coming wide.
Newly settled sponge.
Yeah, right there is the very bottom. You can see the.
There's also a mushroom.
All right, wide.
What do you think the yellow is?
Wide.
Steve?
Plexaurid. Yeah. We have a collection of that from the other site. As far as I can tell, it's the only plexaurid we've seen. I thought there would be more species, but as we go shallower, there will likely be more. On future dives, if we manage to get above 1,000, they should explode in diversity.
How do they look different from that one? Are they different colors?
Different colors, different going.
Oh, this thing.
Yeah.
When will you stop?
Wow.
There you go.
Nice.
Cool.
Susian, right?
Very.
Yeah, it's different colors, different polyp shapes, different colors, and the most common form color is probably yellow, but green and red and purple in some cases. Plexaurids are around.
Awesome.
But yeah, they're tough to tell because the morphology is super variable within that family. And so oftentimes we'll have to actually look at the sclerites that make up each polyp or part of the tissue to refine the ID.
Fish in that cave. Zoom in.
Yeah,
Is there?
cool.
Oh yeah.
You want to get it?
Yeah. Go ahead.
Zooming in.
Mm-hmm.
Cusk.
I think it's a cusk, yeah. Maybe.
That one looks like it has like a cartoon puppy dog eye with
Or
the white glint.
it might be a bifid. Oh, looks good though. Fine looking fish.
Nice zoom.
Fine looking
Yeah.
fish.
You're doing great, fish. Enjoy your cave.
Keep doing what you're doing, which is nothing.
Yeah. Whatever you're doing, it's
Strictly
working
chilling.
for you.
Don't worry, you're protected.
You're in the little hole of safety.
Yeah. We'll leave, so you don't have to freak out. Oh, he's scared one of those decorator crabs up.
All right, I'm moving along now.
Yay. Photobombed.
Did you bump in there, Wayne?
Oh, yeah.
Thanks.
Photobombed by a cheerleader crab.
Little fish there. something.
Mm-hmm.
Sorry.
What do you think? This will be the last step? We'll drift in for a little while and then set it for recovery.
I think we might have time for one more.
Okay.
I'll have to, I haven't had time to do it yet, but I have a more detailed guide than the one that I printed out for the back row with all of the different morphospecies. Oh, can we zoom in right there? Right center screen? There was a red thing.
I see it there, yeah.
Pinkish.
Let me
And I'll have to see if I have a better ID already, or a collection of this yellow fan that I can let everyone know.
Okay, go ahead,
more
zoom.
Better idea of what it could be.
Yeah, that'd be
Stock
great.
crinoid?
Oh yeah, haven't seen too many of these. They were all over the place last time.
Neat.
That's small too.
Yeah, big stock relative, big thick stock relative to the tip of the animal here.
Mm-hmm.
Hmm. Yeah.
Oops. Ooh, it was down there.
Coming wide.
All right, come wide, please.
Coming
Move
wide.
along.
So Steve, do you have a like a
Wide.
species wish list? Are you keeping a total list as we go? Sort of in between dives, what's your process as far as reviewing what we have
I like
seen?
how clear this water is.
Yeah, I mean, it would be nice on every dive to collect one of everything, but I don't think it's necessary because we do have samples from this area of some things, and you know, if we see the same thing on each dive, it's not necessary to collect the same thing at two different sites that are only like 30 miles apart.
Okay.
So I'll try and minimize that impact. But, you know, we do have collections from other regions of the Central Pacific, and so these are great comparisons for those species as well to see if we're looking at something different. Ooh, there was a big puffball something in the lower
Oh,
right.
I see it.
Oh,
It's like a dandelion.
yeah. I think it was Iridogorgia, but super faint.
Yeah.
Have to watch the replay.
Mm-hmm. That's all right. We'd expect to see them at this depth. We saw them on the other sites. We actually saw one that was a couple meters long, 2.5 meters, I would estimate.
Wow.
Big,
Wow.
big colony with a bunch of squat lobsters, shrimp.
They're so beautiful.
We saw many on our watch too, right?
Mm-hmm.
Mm-hmm. Steve, do you have a favorite coral? Do you have a top?
It's like
do you have a
choosing a favorite child, right?
It's easy for you.
How many? Yeah. How many children are you choosing between?
They don't love you back, so.
Yeah.
The corals or the
Oh,
children?
The
ouch.
corals. A coral.
You don't think they love Steve back?
And he
Probably
comes
not.
and terrorizes them every time he sees them.
Let's look at this fish. Zoom in, Wayne.
Zooming.
I'm going to keep coming closer to it as well.
No, I mean, I don't really have a favorite equal opportunity coral biologist there.
Is that the conger eel thing?
No, I don't know what that is. I think, yeah, I don't know what that is. It's not a congrid, for sure it's not. We saw one of these on our watch too, and it kind of just barely got a glimpse of it.
I just want to see your face turn. Hey, heard me.
There's a few scientists out there that do fishes, study fishes, that thought it would be a great idea to make a guide to the fishes based on butt shots, because that seems to be how you get them most of the time.
Oh.
Funny. Yeah, they have enough sensory capacity to try
They're
and
always
get away.
facing away, right?
Mm-hmm.
Yeah, that's a good idea.
there's another dandelion.
Yep.
Yeah, we got time for a zoom on that.
I think.
Missed the previous one,
Ship's
so
holding.
roger that. Okay, Wayne.
Okay, pushing in.
Is that a mushroom? No, that's not a mushroom.
It's a sea pen.
That's a bit different than the one we saw, I think. But
Is that another umbrella or?
That's umbellula, yeah. Mm-hmm.
They're so cool. Look at their little guts.
Yeah. The stalk is actually a modified polyp.
Oh, wow.
Yeah.
Hmm.
Very nice. Yeah, you can see the gut cavity, the darkest region.
Mm-hmm.
That's where the food goes and
Cool.
slowly digested.
All right, come wide.
come on.
Do you have a suggestion of Bazazetus from Neophytidae for the fish?
Yeah, it could be. We do have that in our guide.
Yeah. Unfortunately, any scientists that are following along through just the Nautilus Live website, if you want to sign your suggestions when they come in, they're not logged in. They all come in anonymously through the website. So let us know if you're out there sending in IDs. Love to acknowledge your help with these.
Nice big one right
Thank
there.
you, anonymous scientist. Does look like Basazetus.
Zoom in, Wayne.
See that
Pushing
one?
in.
Another sponge.
Very cool. It's like
It's a flat
kind of
one.
gunmetal gray instead
Very
of being
thin,
white
yeah.
light.
Yep.
Oh, lost
Yeah,
it.
so this is a phenomenon that we don't fully understand, but this is totally normal for sea stars like that to do that, and we don't know why.
Just to bail.
In some cases, yeah, when you're
Come
going
wide.
by with the vehicle in a particularly dense field of corals, it's just
Come
like
wide.
pew pew pew, like they're all just dropping off the colonies, and I have no idea why they would do that. It takes so long and probably so much work to get up to the top of one of these things.
I imagine
They have light sensitivity?
Yeah.
You think so? Yeah,
It would have to be.
I thought it would be like a current associated sensation where if they get the bow wave of
Oh,
the
yeah.
vehicle, that maybe it signals them to, hey, there's some current coming, maybe it'll take me away to a new place.
Mm-hmm.
Oh, yeah.
I'll ride that, but yeah, so that coral to the left there, that's the one we sampled at the beginning of this watch.
Oh, yeah.
I think,
We can get a zoom on it if
yeah,
you'd like.
quick
No problem. Go ahead, Wayne.
Okay, zooming in.
snap. No, it's a bamboo coral. So not the one we sampled, but yeah, this is probably related to those super long ones, or it's the same species as those super long ones we've been seeing.
Is that a crinoid in the background there?
Yeah, it's a stalked crinoid, yep.
Oh, the small one behind it, yeah.
Mm-hmm.
Oh, yeah.
And to the right, there's another.
Everywhere.
Little
What's
anemone.
little orange thing there, yeah.
Yeah.
Yep.
All right, come wide.
Come on.
How much further do you expect Argus to go?
Oh, big fish too. to
Not
the
too
right.
far.
Has it been still sitting kind of like here-ish?
Yeah, I'd expect that. Maybe another 10 meters, but not much.
Okay. All right, so this is the last hurrah.
Mm-hmm.
This is the time when we get any more samples or zooms or whatever. See anything, shout it out.
Mm-hmm.
Little plexaurid again. Yellow one.
Another change in terrain here. It's a little bit more bouldery, a little bit more verticality.
Mm-hmm.
Still pretty crusty, right?
Yeah.
Yeah. Depth now is 1536.
It's fine. Everything's fine.
Mm-hmm.
Nice little golf ball sponge there.
Mm-hmm.
That's actually really big.
Onion, spongian.
Onion spongian, yeah.
It's tough to tell sometimes when they're low to the floor like that if they're actually stalked or not.
Mm-hmm.
Oh yeah, it could be a stalk nestled into the crack or something.
Mm-hmm.
Interesting.
We've actually seen them on other dives totally growing within the cracks of the rock and caves.
Interesting.
Which is a, interesting strategy for a filter feeder.
Maybe that focuses the flow or something?
I have no idea. I mean, you'd think they would want to be out into the flow and get more flow, but it also maybe protects them from,
Oh, right.
extremely strong currents that could dislodge them.
What's that little pink thing right by the lasers?
Pink thing.
Little red thing.
Do you see the
6 o'clock?
orange anemone?
Yeah, I think orange.
You can zoom in there.
Let's give it a go.
Oh, jeez, didn't even see that.
Oh, it's not a... oh, yeah.
Oh, it's a homalid crab.
Homalid crab. Backpack crabs or homalid crabs?
Yeah, backpack crabs, yeah. Yep.
All right, come wide.
Okay, coming
Haven't
wide.
seen any homalids yet with corals. They've all had anemones.
Mm-hmm.
Even though some of these corals that we see, the yellow ones especially, have been seen with, in the backpack or parasol of some of these homalid crabs.
Wayne, bump in, please.
And it's really weird, too, because sometimes you find in the homalid crab branches bits of coral that you haven't seen yet on the dive at all. So you wonder where they come from.
Yeah, where'd you get that?
Okay, so any last samples or anything, or should we get set up for recovery here?
Yeah, so we're gonna in the midwater,
here,
20 meters.
Standing up like this.
20 meters
Let's
up?
say 20 meters off the bottom, take
Do
a
you
Niskin.
mind if I zoom in and we just scan it close up?
Sorry, let's hear what Steve's science goals are first.
Okay.
Yeah, just 20 meters off the bottom with the bottom no longer in sight. We'll take a Niskin and call that a background water sample, and that's it.
Okay.
Okay. Sure, you can have a quick zoom, Wayne.
Okay.
Zoom into nothing, Wayne. Or is it
these
nothing?
are the little tracks. Yeah.
All right, Jake, could you spin to starboard, 180 degrees?
Yeah, it could be a couple things. It could be
Or 030, I guess would be fine.
small mucousy worm tubes, or it could be foraminiferan tubes. Or it could just, these things, you find them all the time on the rocks, and you scratch them gently to see if there's any living tissue inside there, and it's just nothing.
Yeah, that's what.
Yeah. So they could be many. They could be very old.
Okay, come wide, please.
Coming wide. Wide.
So for our environmental DNA, we need a background sample, so a sample that's not directly in coral DNA rich area, just to compare. So you'll sequence the DNA that's collected from this background sample, and then sequence the DNA from within the coral colonies, and then you're looking for additional DNA components that would be indicative of the different corals or the coral mucuses.
All right, you can keep constant depth now. We'll come up with the same delta depth of about 0 for the recovery. Steve, when I was doing that last zoom there, I was pretty close to the front bumper, but my back end was about 5 meters off the bottom. So I'll come up to about 25 meters, which should be 20 to 25 meters off bottom altitude.
Yeah. All right. Yeah, I think the requirements for a background sample are pretty limited. You just don't want to be inside like a massive, garden.
Understood, yeah.
Mm-hmm.
I am really excited to see our cookie star come up.
Yeah.
Okay, you can hold that depth now.
So we'll send a photo of that to some of our shoreside colleagues, and maybe we can get an idea on that even in the next couple days.
That would be awesome.
This week is the 4th of July week, right? So a lot of people are going to probably be taking off for... You're planning on taking off a few days this week?
Yeah, I'm going to go boating.
I hear the Central Pacific's nice this time of year.
I'm working for the overtime.
I'm looking for some good company, though, but may not find it here.
Aw.
Harsh, back row.
Full wide.
I thought we were besties.
Oops. All right, Niskin, anything but the bottom one.
Yep. Yeah, we took the bottom one on our watch.
You took it.
We took 12. We fired 12.
This is the last Niskin of the dive?
Yep.
That's it.
Can I just do a random one, like 8?
Yeah, whatever.
Close your eyes and do it.
I will not do
She
that.
was just like, what up? What? There it is. Got him.
Oh, that was quick.
That's why I wanted that one, is it's easy.
Was that 8?
Yeah.
Oh, I see. Okay. Yep.
Okay. Let's
Thanks,
get
Steve.
nice and stretched out here, and then we're good to go.
Pumping in. Wide.
Okay, you can come out of auto heading, and you can even secure your thrusters. And then you'll start coming up on a winch, and you might want to use the stick.
Okay.
We'll start at 12, 15 meters a minute.
So
You might adjust it from there. All right, that's looking good. So now I can start this action here. So let's talk through a recovery. So we're going to keep coming up. Right now I'm lateraling around to spin around, so I'm facing it that way we talked about earlier. My heading opposite the ships here, and I'll end up pulling your heading around, so yours will match the ships.
Okay.
You can speed up a little bit to 18. Maybe even 19. That's doing pretty good. Maybe even 20.
Flooring it.
Yeah, so once we... yeah. Good?
Yeah.
All right, cool. Yeah, so we'll keep coming up. Once I get streamed out there, we'll look pretty good. keep coming up. Not a lot